Hi there. In this first of 3 videos on DNA sequencing, we're going to talk about conventional DNA sequencing, and we're going to focus on the sequencing reaction itself. This type of sequencing goes by several different names. It can be called the Sanger method of sequencing, it can be called dideoxy sequencing, or it can be called chain termination sequencing. We're going to look at the key role of the 3' hydroxyl group on the bases for this method to work. We're going to show you that the fluorescent color of the base is going to indicate which nucleotide is present, and that the product length of the sequencing reaction is going to indicate the position of the nucleotide. There are a number of good online resources for sequencing, and since the human genome was sequenced in 2001, 10 years later there was a special report published about the progress of the human genome, and this is publicly accessible at this URL. It's interesting to check out if you are so inclined. Now this slide in a sense summarizes the next 3 class sessions, what we're going to discuss. You'll remember the central dogma of biology is that DNA makes RNA makes protein, that these different molecules are specified by strings of deoxyribonucleotides or ribonucleotides or amino acids. And the high throughput methods for studying these 3 different types of biopolymers will be DNA sequencing for DNA obviously, microarrays and also RNA sequencing for the study of RNA, and mass spectrometry for the study of protein. Like I said, we'll be covering each of these biopolymers and methods to study them in detail in the next couple classes. So the goal of DNA sequencing, which might be rather obvious, is simply to find the complete sequence of A, C, Gs, and Ts in DNA. And sort of at the pinnacle of Sanger sequencing in the 90s and early 2000s, this kind of data was obtained on benchtop machines like this, and the data ended up looking like this for about 600 to 800 bases, with different peaks indicating the bases that predominated that position. Of course, what you might be aware of, and we'll see in the 3rd video today, is that sequencing has undergone a revolution that even surpasses Moore's law. Moore's law is in reference to the number of transistors that are present on an integrated circuit, and that it doubles approximately every 2 years. And this has held true roughly since the early 70s. So for almost 40 years now we've enjoyed exponential growth in transistor number packed onto integrated circuit, and despite numerous warnings that we're about to reach a fundamental limit, people keep innovating and increasing the speed of our processors. Progress in DNA sequencing technology has outpaced Moore's law, and this is evidenced by a drop in price for sequencing a human genome. In 2003 it's about $1 billion, and in 2010 you could already get a company to sequence your genome for about $5,000, and the price has probably dropped since. But this represents approximately a drop in a factor of 10 for every year that technology is allowed to evolve. So if you project this out, you are going to hit a limit. DNA sequencing will be free, and it can't get any cheaper than that. So you might want to start thinking about what the uses are for sequencing, because it is the single most powerful tool to understand biology. So in this slide we highlight some of the milestones in DNA sequencing: discovery of the double helix, development of recombinant DNA, the first publication by Fred Sanger of DNA sequencing with chain termination inhibitors that we're going to see. That's why we call it Sanger sequencing. Then in '87, the development of automated DNA sequencing machines. Up until that point, it's DNA gels, which people poured by hands. Now the method is automated. Then in 1995, the first genome of a bacteria is sequenced, Haemophilus influenzae, by Craig Venter and his team. In 2001, the human genome, a draft sequence of the human genome, is published. And then in 2004 and 5, we get into the next generation sequencing where the costs plummet rapidly. And here's the molecule that's the key for this method. Here is a standard deoxynucleotide. Remember, it's deoxy because RNA has a hydroxy here, but it's deoxy. We just have a hydrogen atom. But we have a hydroxyl group at the 3' end, and you know that that is where the growing DNA chain will attach to. But in a dideoxy, you have no hydroxyl here and you have no hydroxyl here. Nothing will be able to add on to that base, so this is going to terminate the chain that's being synthesized. And now we'll see how that can be used to read out a sequence of DNA. And again, here we're just highlighting a chain of DNA growing from the 5' end down to the 3' end, and then you have the nucleotide triphosphate attacking the 3' hydroxyl to form a new bond. If that 3' hydroxyl isn't there, there can be no attachment of another nucleotide. So when we're doing sequencing, this is the strand that we're trying to sequence. This is the primer that we're going to use to start the synthesis. We're going to use the enzyme DNA polymerase. We're going to have a pool of nucleotides, and we're going to add, again, growing this strand, 5' to 3', we're going to add the bases that are complementary to the existing strand. Every once in a while, we're going to hit a dideoxy, and it's going to get incorporated, and that's going to terminate the chain. We won't be able to add any more bases. Now, if we look at the nucleotides that are included here, most of the A's are normal deoxyribonucleotides, but occasionally you have a dideoxy, so you have a little bit of this chain terminator mixed in with plenty of regular nucleotides. These will get incorporated because you don't want to, if you only had the dideoxys, the first time you hit an A, you would stop. This allows you to, with some statistical probability, read through every base, but occasionally you will stop at a particular base and then you will have a fragment. And just to make it explicit, these are the 5 things that are included in a sequencing reaction. Now, if we do this sequencing reaction with a little bit of dideoxy-A's mixed in, a little bit of dideoxy-G's, a little bit of dideoxy-T's, and a little bit of dideoxy-C's, and they are colored in different colors here, and that's because each of the dideoxys have a fluorescent molecule attached to them. And so whenever they get incorporated, that strand is going to appear fluorescent with a different color. So there's 4 different colors because there's 4 different bases. Now in one sequencing reaction, we've got many copies of the template molecule, millions or so. We've got a primer that's going to sit down on every single one of these, and in some cases we're going to terminate eventually on an A. Sometimes we're going to terminate on a G, so it's all mixed up into one soup. And what you'll notice is that these different fragments are different sizes. Sometimes it terminated early, the first time an A was encountered. Other times it put in normal A's, and only on the 3rd occasion put in a dideoxy. So when we interpret this data, we're going to read the color to indicate which base it is, and we're going to look at the size of each of these fragments and figure out the position. So we're going to run that out on a gel. As you know, if you put the DNA in here, and this can be done in one lane, one for each of the bases, or I'll just jump ahead. You can put them all in a single lane, but let's look at it here first. You're going to put these bases in here. This is the A band. The smallest one is going to move the fastest because it's migrating down. So here's that fragment. And then you're going to be able to read the bases right off the gel. A, T, G, T. If we look at what we started with and where the primer is, so that's right here, and it's going to be A, T, G, T, and so on. So you'll be able to read up the gel to figure out what base it is. But a key here is that we're able to separate out these fragments with single base pair resolution. This band is 5 nucleotides, this band is 6 nucleotides, and so on. But since the bases are actually different colors, we can run them all in a single lane simultaneously. And now you can see very clearly that the color gives the base, and the position on the gel, so the size of the fragment, gives the nucleotide. So here we see green, pink, blue, yellow, green, pink, blue, yellow. And in this way we've done a sequencing reaction. And this is what data looked like in the mid to late 90s. and these when we were still using sequencing gels. Here there are approximately 32 sequencing lanes, and you can see that if you go down any one of these lanes, you would be able to read the colors and deduce what base sequence is there. But pouring gels are kind of tricky, and as you can see, there's a little bit of a bending here, which happens because of differential heating across the gel, and you could sometimes have bleeding from one lane a little bit into another lane, and so people looked for a more automated system, and what they came up with was capillary sequencing, and that here's a capillary, and inside this capillary you have an agarose material, just like the agarose that's in gels, and you're going to put samples in here, and they're going to migrate under electrophoretic bias, and the smaller pieces will migrate faster, and they're going to all pass a particular location where there's a laser sitting, and that's going to detect the fluorescence. So we're not reading the fluorescence at various points along this agarose, I'll call it a gel, but we're going to just look at different times what comes past here. And what's being depicted here in terms of how you observe it, you've got a complex mixture of nucleotides here, and this is the place where you're measuring the fluorescence, and then they're going to electrophorese. Here you can see the smallest molecules are the green ones, followed by blue and red, and then a little later time the red has finally gotten into this fluorescence zone, and eventually I guess it looks black, green, and blue will all pass through the detector, and you'll read off the fluorescence just like you did on the gel, and you'll depict it in this way, which shows the traces for all the different colors simultaneously. One of the great advantages, well, several of the great advantages of this capillary system rather than gels is gels had to be poured by hand. They were prone to errors. This capillary material, once a reaction was done, the agarose inside could be expunged automatically. It could be refilled by the machine. You could do another sequencing reaction, leading to a lot greater speed and a lot greater reproducibility. Plus they went from 32 lanes in a gel to 96 capillaries. So if you are uncertain about how the sequencing reaction works, I highly recommend this video. It's only about 2 or 3 minutes. It is extremely succinct and it will help you visualize the sequencing reaction, and there's a link on the class homepage to this. There are also links on that page to a series of animations put out by the Department of Energy that will describe many aspects of DNA sequencing, as well as 2 articles which you can use for reference, one on sort of conventional sequencing and then the latest on the next generation sequencing. Okay, thank you for listening.